Hello students, hope you are enjoying and studying well at your home. In the previous video, we discussed about the pollination and agents of pollinations. Now we will discuss about the process or events that happens after pollinations. When different agencies or transfers the pollen grains at the stigma, then stigma start identification of these pollen grains. Like in the previous sections, we discussed that the plants which favors self-pollination. They do not allow the another pollen grains to germinate over there. And the same case, the plants or flowers which allow only the cross-pollination, in this case, the self-pollen grains are self-incompatible. Means the pollen grain, when they reach the stigma start identifying the pollen grains, that is it the right type of pollen grain or it's a not. मीन्स जब पॉलन ग्रेन स्टिग्मा पे पहुंचते हैं तो स्टिग्मा अब आइडेंटिफिकेशन करती है क्या ये पॉलन ग्रेन सही टाइप के पॉलन ग्रेन है या नहीं. अगर सेल्फ इनकम्पेटेबल पॉलन ग्रेन होंगे देन दे आर नॉट अलाउड टू जर्मिनेट देयर इन द केस ऑफ सेल्फ और क्रॉस पॉलिनेशन. Same thing happens in the case of cross pollination when cross pollination allowing flowers do not allow their own pollen grains to germinate on the stigma. These events after pollinations, these events happens, it starts from the pollen pistil interaction. Pollen pistil interaction is the events in which we include all the processes which starts from the reaching of the pollen grains at the stigma to the entry of the pollen tube inside the ovule. All the events included in these are known as pollen pistil interaction. Now let's see what happens there in the pollen pistil interaction. Pollen pistil interaction. These are the events after reaching the pollen grains at the stigma till they form the pollen tube and it enters inside the pollen grain. All these events are known as pollen pistil interaction. Let's see. When pollen grains, suppose this is the stigma. We're just drawing the stigma part. Suppose this is the stigma and pollen grain reach at the stigma. This is the pollen grain which have intine as well as exine. It is formed from the two layered structure. Two layers are present here and these layers are not in the continuous manner. It has some pores in between these and these pores are known as germ pore, about which we have already studied. It's exine and intine. Two layers are present on the pollen grains. Intine is the continuous layer and inner layer, and exine, which is the outer layer, and it's not continuous. When these pollen grains reach at the stigma start identifying it or it's start rejecting or identifying it. Means वो देखेगी क्या pollen grain सही टाइप का है. अगर सही होगा तब आगे की सारी इवेंट्स जो है वो स्टार्ट होंगी. तो फर्स्ट स्टेप इज identification. First is identifications of pollen grain. First thing that happen here is the identification of pollen grain. If the pollen grains are of right type, then the interaction or next process will start. What is the next process? After the identification, now this pistil, or we call it pollen pistil interaction. First, let's see why we are saying that pistil. Because pollens are not directly interacting with the female gamete. यहाँ पे pollen grains directly female gamete के साथ interaction नहीं कर रहे हैं, क्योंकि female gamete जो है वो कहाँ present है, वो यहाँ नीचे ovary और ovary के अंदर ovule में embryosac के अंदर present है. It's very far from the pollen grains. First of all, pollen grains are just interacting with the pistil. It's just interacting with the pistil. That's why it is known as pollen pistil interaction. First, pistil will identify the pollen grains and then let's see what happens. After that, this pistil or this stigma part, the stigma starts secreting. It secretes sugar substance. It will start secreting sugar substance. When pollen grains reached and they are identified as a right type of pollen grain, then pistil or this stigma part will secrete a sugar substance. This sugar substance contains water and sugar. Here it secretes sugar, in which water is also present. It secretes this sugar and water, means its sugary substance is secreted by the pistil. After that, this pollen grains will receive or this absorb this sugary substance. They will absorb this sugary substance, and after absorbing this sugary substance, the intine now start forming a tube-like structure that is known as pollen tube. As you know that enzyme is the hardest substance. It's not able to get decomposed. ये तो खत्म होगी नहीं, ये तो इसका तो कोई इफेक्ट होगा नहीं इस पे शुगर सब्सटेंस का. सिर्फ इंटाइन है वो pollen tube formation का काम करेगी. बट एग्जाइम्स जहां पे कुछ हमने देखा था कि कुछ गैप हमने छोड़ा था जिन्हें हम बोल रहे थे germ pore. So pollen tube will start forming only at the germ pore levels. Let's see how it will start. This is the pollen grains. This sugar substance will release here and it is absorbed by intine and it start forming a pollen tube. If we discuss the structure that we already discussed, the structure of pollen tube, structure of pollen grains, it has two types of cells. One larger size cell is known as vegetative cell, and the smaller spindle shaped cell is known as generative cells. This is the generative cell. And this larger size cell in which the generative cell floats is known as vegetative cell. Vegetative cell or tube cell. We can call it tube cell also. This is the two-celled stage. These the two cells combinedly form the two-celled stage of the pollen grains. Most of the flowers, two-cell stage. Most of the pollen grains are released in the form of two-celled stage. So we are discussing or we are taking the same example that pollen grains are here in the two-cell stage. जैसा हमने पहले देखा था कि जब pollen grain रिलीज होते हैं तो वो टू सेल स्टेज में ही रिलीज होते हैं. After that they will form the three cell stage. Here we are taking two cell as a similar example. It's a two cell stage in which one cell is vegetative cell and another cell is generative cell. Now let's see what happens there. First, sugary identification. Pollen grains are identified. Then stigma secrete sugar substance. This sugar substance is absorbed by these pollen grains or the intine of the pollen grains. Now pollen tubes start forming. Next process or next event that will happen here is the pollen tube formation or pollen tube start forming. Now pollen tube will form after when these intine will absorb the sugar substance. It will start germinating and they form a pollen tube. In some flowers or in some plants, one pollen tubes are formed, or in some another flowers, more than one pollen tubes can be formed. It depends on flower to flower, but only one pollen tube will enter inside the stigma. Only one pollen tube will enter inside the stigma. When only one pollen tube is formed, then this condition is known as one pollen tube is formed, that is known as monosyphonogamy. Monosyphonogamy. When one pollen tube is formed, then it is known as monosyphonogamy. Mono means one, then only one pollen tube is formed. When more than one pollen tube are formed, more than one, then it's known as polysyphonogamy. So here we see two type of condition, monosyphonogamy and polysyphonogamy, when one pollen tube forms and when more than one pollen tube is formed. After the germination of the pollen tube, they start forming pollen tube. pollen tubes now start entering inside the style part. This is the style. Now, when pollen tube, we discussed the 3 points. The fourth point will be entry of pollen tube. Entry of pollen tube in style. Now, pollen tube will start moving inside the style. When pollen tubes start moving inside the style, all the content of these cells are now released into this pollen tube. We will draw it more clearly so that we can easily see it. This is the pollen tube, which forming from these pollen grains, and all the contents of these pollen tubes is released inside this pollen tube. When this pollen tubes start forming, at that time the generative cells get divided. At the time when pollen tube starts entering inside this style, at that time generative cells divide and it forms two male gametes, and this is known as three-celled stage. So we will have to make two gametes here. One is one male gamete, second is another male gamete, and with behind this all the cytoplasm of vegetative cells is also present with its nucleus. So what happens here? Let's see. Now it's empty. Here nothing is left behind here. What happens when pollen tubes start forming? It enters inside the style. Pollen tube बनेगी और वो स्टाइल के अंदर एंटर करेगी. जैसे pollen tube स्टाइल के अंदर एंटर करती है, उस टाइम पे वेजिटेटिव सेल और जनरेटिव सेल पहले डिवाइड होके टू मेल गैमेट्स फॉर्म करने का काम करेगी. Now pollen tube will come into the three-celled stage. What happens next? Then this pollen tube will come inside this style, and all the content of these cells are entered inside the or released inside this pollen tube. सभी का सारा पैटर्न जो है इस सेल्स इस pollen grains का वो इस pollen tube के अंदर रिलीज कर दिया जाएगा. This happens so far. Now this pollen tube will enter inside this style. Now style is the part, long tube-like part which connect the stigma with the ovary. This style can be also two types. It could be hollow type or solid type. Let's see what happens. Style could be hollow type. Hollow type style means it has only the epidermal cell. अगर style में सिर्फ epidermal cells present है और अंदर का सारा portion खाली है, तो उस style को हम बोलेंगे hollow style. Then style is known as hollow style. In solid type of styles, all the contents, the inner content is also filled with parenchymatous tissues. अंदर की तरफ पूरा का पूरा जो एरिया है, that is also filled with parenchymatous tissue. Such type of style is known as solid style, and these tissues are known as conductive tissue. They form the conductive tissue. Now let's see. अब तक हमने क्या देखा है, एक बार फिर से रिवाइज करते हैं. हमने देखा सबसे पहले पॉलन ग्रेन जब स्टिग्मा पे पहुंच रहे हैं, स्टिग्मा पे पहुंचने के बाद सबसे पहले आइडेंटिफिकेशन होगा दैट इज द पॉलन ग्रेन्स आर राइट टाइप और नॉट. इफ द पॉलन ग्रेन्स आर ऑफ राइट टाइप, अगर सही तरीके के होंगे, तब स्टिग्मा शुगरी सब्सटेंस रिलीज करता है. उस शुगरी सब्सटेंस को अब्सॉर्ब करके पॉलन ट्यूब की फॉर्मेशन स्टार्ट हो जाती है. व्हेन पॉलन ट्यूब स्टार्ट फॉर्मिंग और एंटरिंग इनसाइड दिस स्टाइल, जनरेटिव सेल गेट डिवाइड एंड इट फॉर्म्स थ्री सेल स्टेज. इट फॉर्म्स टू गैमेट्स. ये जनरेटिव सेल टू मेल गैमेट फॉर्म करेगी. नाउ ऑल द कंटेंट ऑफ दीज पॉलन ग्रेन्स आर नाउ रिलीज्ड इनसाइड द पॉलन ट्यूब. मींस दोनों मेल गैमेट, वन मेल गैमेट, सेकंड मेल गैमेट एंड द साइटोप्लाज्म एंड न्यूक्लियस ऑफ वेजिटेटिव, सबका सब इस पॉलन ट्यूब के अंदर रिलीज कर दिया गया है. अब पॉलन ट्यूब इसके अंदर स्टाइल के अंदर मूव करना स्टार्ट करेगी. दिस इज द स्टाइल. इट्स अ सॉलिड टाइप ऑफ द स्टाइल एंड इट स्टार्ट मूविंग इनसाइड द स्टाइल. नाउ लेट्स सी व्हाट हैपेंस when pollen tube move inside the style. So all the content, now let's see, we are drawing pollen tube here. It has one male gamete, second male gamete, and all the material or the cytoplasm of vegetative cell also is there. When this pollen tube enter inside this style, all the content do not get back. पूरा का पूरा जो content है pollen tube का, वो वापस पीछे की तरफ move ना कर जाए. इस चीज को प्रोटेक्ट करने के लिए यहाँ पे कैलस फॉर्मेशन होता रहेगा. यहाँ पे एक तरीके की एंट्री ब्लॉक की जाएगी ताकि सारा कंटेंट पीछे की तरफ मूव ना होता रहे. धीरे धीरे pollen tube move करेगी and by by entering, it will start entering inside this style. It will move inside the style, and all the content will also move with the pollen tube inside the style. This is the and then it will move, and finally it will reach. Let's see what happened. Finally it will reach at the ovary. Here we can draw ovary. When pollen tube is moving, it will enter inside the ovary, and inside the ovary here the ovules are present. Ovule जो है वो अंदर present होगा, और ovule के अंदर present होगा embryosac. कैसे अब pollen tube की entry अंदर होगी, कैसे embryosac तक pollen tube पहुंचेगी, इस चीज को हम अभी कंटिन्यू करेंगे in our next section of the video. Now let's see what happened so far. We've studied that how the pollen tubes are formed, how all the contents of the pollen grains are enter inside the pollen tubes, and how pollen tube will move inside the ovary. And now next section we will discuss about the entry of the pollen tube inside the ovule. So keep watching and keep studying. Thank you.